Thank you for the invitation to participate in this microbiology virtual week. Coronaviruses are a family of viruses that infect vertebrate animals like birds and mammals. In wild and domestic animals, coronaviruses mainly cause enteric and respiratory diseases. In humans, they are associated with respiratory infections. Common ancestors of different coronavirus genera have been found in bats living in all continents. From bats, coronaviruses cross the species barrier, first to intermediate hosts and then to humans, that may become a pandemic. The presence of coronaviruses in ubiquitous animal reservoirs makes the possibility of new pandemics in the future a realistic option. At present, 7 human coronaviruses are known. The 4 in yellow cause mild respiratory infections like common colds. Some of them, 229E and OC43, are known since the 60s. The other 3 coronaviruses in red have emerged in the 21st century, and all of them cause pneumonias that can be deadly in humans. The first SARS coronavirus emerged in China in 2002 from civet cats as the intermediate host. It caused an epidemic with 8,000 infections and 800 deaths. MERS coronavirus emerged in 2012 in the Middle East and has been causing sporadic infections from dromedary camel to humans with a mortality around 30%. SARS-CoV-2 has caused the current pandemic, and its intermediate host has not yet identified. For the moment, more than 4 million deaths have been caused worldwide. The possibility of emergence of new coronaviruses makes necessary to improve our preparedness against future pandemics. Coronaviruses are positive strand RNA viruses with one of the largest known RNA genomes of around 30,000 nucleotides. The genomic RNA associated with the nucleocapsid protein in red is protected by an envelope that includes different structural proteins. The membrane protein, or M, the envelope protein, or E, and the most prominent, the spike protein, or S. S protein is responsible for the binding to the cellular receptor, and it is also the main inducer of neutralizing antibodies. For this reason, S protein is the only component of most of the current vaccines against SARS-CoV-2. Different vaccine strategies against SARS-CoV-2 have demonstrated to be safe and efficient. Inactivated viruses are obtained with traditional technologies. They have limitations in terms of efficacy and might have safety problems if inactivation is not complete. Using biotechnology, other vaccines consisting either in subunit proteins or in nucleic acids have been engineered. Recombinant proteins are easy to product, and they usually need adjuvants to increase efficacy. Viral vectors usually adenoviruses, have been engineered to express this S protein from its genomic DNA. They do not replicate in the cells and they are attenuated. These vaccines have some advantages, such as the lower cost of production, and they are easy to store and transport. mRNA vaccines are represent the most innovative technologies. They include the synthetic mRNA coding for the S protein, which is the minimum information to produce the antigen inside the cells. The mRNA is protected and delivered using lipid nanoparticles. These vaccines have demonstrated to be safe and highly effective in the real world. However, they have some limitations, such as the high cost of production and the requirement of ultra-cold temperatures for storage and transportation. Although the correlates of protection for SARS-CoV-2 are not completely known, there is some information from convalescent patients about the conditions of a protective immune response. IgG and IgA neutralizing antibodies against the S protein have been found in almost all the patients, in addition to a cell response against the S protein. Interestingly, CD4 and CD8 T cells specific against epitopes present in M, N, and non-structural proteins were also found, indicating that other viral proteins in addition to S protein are also required to get a protective immune response. mRNA vaccines have important advantages. First, safety. mRNA cannot be integrated into genomic DNA, so there is no potential risk of mutations. mRNA is degraded by cellular RNases. Its half-life in vivo is short, so there is no risk of toxicity by overexpression of the protein of interest, the S protein. They contain a few chemically defined components, which simplifies the regulatory process. RNA vaccines have demonstrated to be highly efficient, and the expression of RNA requires optimization of stability and translation. Carrier molecules are required for the efficient in vivo delivery of mRNA. Although large-scale production of mRNA vaccines is feasible, its more economical options should be welcome. In addition to the current non-replicating synthetic mRNA vaccines, virus-derived self-replicating RNAs represent promising next-generation vaccine candidates. They have been optimized by viral evolution in terms of replication, gene expression, and evasion from the innate immune response. Today, I will present our work in the development of self-replicating RNA vaccine candidates against SARS-2. In our laboratory, we have been working in coronaviruses for more than 35 years. We are interested in the identification of virus virulence factor. Deletion of these genes from the viral genome leads to attenuation, and attenuated viruses are vaccine candidates. On the other hand, the identification of viral and cell pathways involved in pathogenesis provides information about potential therapeutic targets for antivirals. Working with highly pathogenic coronaviruses requires biosafety level 3 protocols and laboratories, as this one shown in this picture. It corresponds to our laboratory at the National Center of Biotechnology in Madrid, in Spain. To study viral pathogenesis and to develop vaccines, we have established two essential tools. First, an infectious cDNA clone. This is a full-length DNA copy of the viral RNA genome, which can be modified in the lab using molecular biology techniques. We were the first lab in the world in the year 2000 to develop a reverse genetic system for coronaviruses, which was challenging at that time because of the large size of the viral genome and because of the presence of toxic regions for bacteria. And second, small animal models that reproduce the severe disease observed in humans. They are critical to evaluate the efficacy of vaccines and antivirals. Recombinant viruses can be rescued from the infectious cDNA clone, which is assembled by joining synthetic DNA fragments. This cDNA clone is introduced in bacterial artificial chromosomes, which are transfected into mammalian cells. RNA polymerases from these cells transcribes the DNA into an RNA molecule which is identical to the genomic RNA of viruses. This RNA starts the virus life cycle, leading to infectious viruses that can be rescued from the supernetting of the cells. Genetically modified humanized mouse models that express the human receptor of the virus reproduce the infection of the lungs, clinical signs, including the inflammatory pathology and pulmonary pathology, and finally, the death. In the last few years, we have been working in the development of MERS coronavirus RNA replicon as vaccine candidates, which are replication-competent and propagation-defective. These results are the proof of principle for the development of vaccine candidates against SARS-CoV-2. Using the reverse genetic system, a deletion mutant of MERS coronavirus lacking the envelope or E protein was generated. This deletion mutant was propagation defective, as shown in the left graph with the red line. This deletion mutant was propagation defective, which means that it was lost during passages in cell cultures, in contrast to the continuous growth of the wild-type virus, as shown with the black line. By providing in trans the E protein in packaging cell lines, the growth of the defective virus is partially rescued. These viruses are replication competent, as shown in the right graph. The red columns show that they produce viral genomic and subgenomic RNAs to similar levels as the wild-type virus, as shown in the blue columns. Since they have lost its ability to propagate, they are not viruses anymore, and they should be considered just as RNA replicons. These RNA replicons are produced from the engineered DNA replicon in packaging cell lines, which provides the missing E protein in trans. Other viral structural proteins are produced from the self-replicating RNA to form virus-like particles that include the viral RNA, the RNA of the replicon, to be delivered to target cells. Inside the cells, RNA replicon amplify its RNA and produce the proteins, the antigenic proteins of the virus, and form these virus-like particles, which are antigenically similar to the wild-type virus shown to the left. This provides an improved immune response because these virus-like particles not only contain the spike protein, but also other viral proteins which are essential to improve the immune response. However, these virus-like particles are defective in propagation because in target cells, the E protein is missing, which makes the replicon viruses highly safe. The attenuation of the RNA replicons was further increased by the deletion of accessory genes. These genes are not essential for viral replication in cell culture, but they are involved in virus virulence in vivo because they interfere with the innate immune response. Therefore, deletion of these genes increases the innate immune response and, as a consequence, also the adaptive immune response. MERS coronavirus encodes 4 accessory genes, 3 for A, for B, and 5. To illustrate the contribution of accessory genes to virulence, the deletion mutant MERS-delta-4B engineered and evaluated in humanized mice for clinical signs, weight loss, and mortality. MERS-delta-4B, as shown in the red line, caused an initial weight loss that was followed by a recovery, in contrast to the severe weight loss produced by the wild-type virus, the black line. As a result, all wild-type infected animals died, as shown to the right in the black line, while animals infected with the delta-4B virus, the red line to the right, all of them survived, indicating that 4B gene was a virulence factor and delta-4B virus was attenuated in vivo. Attenuation of MERS-delta-4B virus was associated with a decrease in the innate immune response, as observed in the red columns corresponding to delta-4B virus. Lower levels of pro-inflammatory cytokines like IL-6 and chemokines were observed in the lungs of mice infected with delta-4B virus as compared to the wild-type virus, corresponding to the black columns, although both viruses replicated to similar extent. In addition to RNA replicons lacking one gene, the E gene essential for propagation, or 4 accessory genes involved in virulence, corresponding to the second scheme in the slide. Another RNA replicon was generated, including a deletion of the two sets of genes. This RNA replicon, lacking 5 genes, was replication-competent and propagation-defective. It was self-replicating because it includes the replication machinery indicated by the blue boxes. And this self-amplifies viral antigens and the antigenic dose, including not only the S protein, but multiple viral antigens, like genes M, N, and non-structural proteins included in the replicase, which improves the immune response. In addition, it is highly safe because it lacks genes essential for propagation, like E gene, and all the accessory genes involved in virulence. The attenuation of MERS RNA replicons was evaluated in humanized mice. In contrast to the wild-type virus, please see the black line corresponding to the wild-type virus, which caused severe weight loss to the left and the death of all the animals to the right. RNA replicons lacking either E gene in green or 4 accessory genes in red or the 5 genes in orange, these mice did not show any clinical signs and all the animals survived, indicating that RNA replicons were attenuated in vivo. The replicon lacking 5 genes, the accessory genes and the envelope gene, was replication competent in vivo, as shown by the orange columns, indicating the synthesis of genomic RNA to the left or subgenomics RNA to the right, although to a lower extent than the wild-type virus in the black columns as expected. This replicon lacking E and the accessory genes was propagation deficient in vivo. As observed in the orange columns, no infectious virus was detected in the lung of infected mice as compared to the efficient production of infectious particles, around millions of infectious particles observed when the animals were infected with the wild-type virus as observed in the black columns. Protection of mice induced by MERS RNA replicon was studied by the intranasal immunization of humanized mice with a single dose of RNA replicons, followed by the challenge with a lethal dose of MERS wild-type virus. As observed in the gray line, non-immunized animals showed a severe weight loss after the challenge and all the animals died. In contrast, in the colored lines, animals immunized with the RNA replicons did not show any clinical signs or weight loss after the challenge and all the animals survived, indicating that immunization with the RNA replicons lacking 1, 4, or the 5 genes provided 100% protection to the immunized animals. Interestingly, the immunization provided by the replicons was sterilizing, as observed in the orange columns, indicating that no challenge virus was detected in the lungs of immunized mice, in contrast to the effective growth of the challenge virus in non-immunized mice, as shown in these black columns. This sterilizing immunity not only protects against the disease, but also against infection and transmission. This is a concept corresponding to an ideal vaccine. Current vaccines provide an efficient protection, especially against disease and death, but they not provide an effective protection against infection and viral transmission. So the idea of getting an sterilizing immunity is an interesting concept for next generation vaccine candidates. Following a similar strategy to that used with MERS coronaviruses, we are currently working in the development of a vaccine candidate against SARS-CoV-2, also consisting in replication-competent propagation-deficient RNA replicons. Starting from the infectious cDNA clone of SARS coronavirus, as shown in the first line, a collection of deletion mutants of SARS-CoV-2 have been generating, and replication-competent propagation-deficient RNA replicons have been obtained by the deletion of a combination of genes which are required for propagation and also involved in virulence. Two formulations are being assayed for the delivery of the RNA replicons of SARS-CoV-2. To the right, the strategy of virus-like particles, similar to that described for MERS coronaviruses. The DNA coding for the RNA replicon is transfected into packaging cell lines, which provide in trans those structural proteins necessary for propagation to generate virus-like particles that include the RNA replicon inside. In target cells in immunized animals, these VLPs introduce the replicon RNA, but in the absence of the proteins required for replication, there is no possibility of generation of infectious viruses, and only the RNA inside the cell self-replicates and produces the antigens necessary for eliciting an effective immune response. The second strategy consists in synthetic cell-free delivery system. The RNA of the replicons is produced by in vitro transcription and combined with polymers to form nanoparticles. In addition to the intramuscular immunization, we are also working in the intranasal administration of these RNA vaccine candidates, because intranasal immunization leads to mucosal immunity, which is essential to provide sterilizing immunity and to get this protection that not only protects against disease and death, but also against viral transmission. And finally, the acknowledgement to all the lab members participating in this project, especially to Luis Enjuanes, who started the coronavirus laboratory in Madrid many years ago, and to our collaborators, and of course to all of you for your attention. Thank you very much.